So good afternoon, everybody, and welcome to this webinar. We're still going to wait for a few minutes to give attendees a little bit more time to join the session. So we'll be starting in a bit. So hello everybody, and welcome to today's webinar about ToxTracker, its applications, and some new essays and tools we will launch today. I'm Paula van Rossum, the Chief Business Officer from Toxis. As you probably know, this program was originally intended as a hosted session alongside the SOT. However, since the SOT was cancelled, we have immediately contacted the speakers to see if they were willing to do the presentations via a webinar, which they did. This also allowed more people to join, and we were very much impressed by the overwhelming interest to join. So thank you all for your interest. I'm proud to share the program and introduce our excellent speakers. The first presentation will be by Jill Hendricks, our CEO, on the developments around ToxTracker and new assays. As our crowd today consists of both users and those who are new to ToxTracker, we will start with an introduction and later show some new developments. So for some, there might be some repetition. After Jill, there will be a presentation by ToxTracker. Jax is a genetic toxicology scientist at RIFM, the Research Institute for Fragrance Materials. He will give a presentation on some of the work that has been done on testing flavor and fragrance compounds. And finally, Raja Setivari will give a presentation. Raja is leader genetic and molecular toxicology at Corteva Agrisciences. Corteva has been part of the validation study, but they are now also using ToxTracker in-house. He will share his experience with the assay. Originally, there was also a presentation by Ruth Dunsizen from the Cobalt Institute. However, due to personal circumstances, she's not able to give the presentation today. After each of the presentations, there will be time for a few questions. Please use the chat function also during the presentations to ask questions. I will monitor the questions and ask them to the speakers afterwards. At the end of the presentations, there will be more time for questions you might have. We will try to answer all of your questions. If not today, then we will get back to you. The presentations will not become available afterwards, except for the presentation by Toxis. So after these household messages, we can start the presentations.
So good morning or good afternoon to everyone. This is Giel Hendricks. I assume that many of you know me. I'm the CEO and founder of Toxis. Also on behalf of me, thank you for joining this webinar. It's unfortunate that it was not possible to see you in person at the SOT conference this year, but I'm happy to see that you took the time to join this webinar. For us, like probably for all of you, these are rather strange circumstances. I am sure that all of you are dealing with the coronavirus outbreak at the moment, probably working from home. And also here at Toxis, we are struggling our way through. Many people are working from home, but the labs are still operational. We still run the services for our clients, and also most of the R&D activities continue, at least as well as possible. So we are managing, but we will do everything in our powers to keep at least our services operational, and we have the capacity and enough overflow with people to make sure that we can run the services that we provide. So as a very quick introduction to those of you who are not familiar with Toxis. So Toxis is a biotech company developing in vitro systems for toxicological research. And we offer these systems or these assays under a license or as a kit, but also as a service. So we also work as a contract research laboratory. And it's fair to say that our main expertise is in the area of genetic toxicology, cancer hazard assessment, and developmental toxicity. We have a client base basically all over the world in different industries, so pharma, chemical, agrochemicals, cosmetics, food industry. We are founded in 2014, originated from the Leiden University Medical Center, but now for a number of years have our own laboratories here at the Leiden Bioscience Park in the Netherlands. We currently have 14 people employed, and so we highly value science, and we are actively participating in various scientific expert organizations. Many of you or some of you might have seen that we recently on the business side also raised a new investment round to further develop the company, expand to the US, work on regulatory acceptance of the assays that we're developing, and of course invest in the development of new assays. And later on during this webinar, I will show you a few examples of those new assays. So a very brief introduction. This is a slide that I am sure that many of you have seen previously. So ToxTracker is a stem cell based reporter system where 6 different reporters, fluorescent reporters, are combined in a single assay that allows the system to detect various toxic and genotoxic properties of compounds. And the advantage of the system is that it can identify a number of different mechanisms of action of genotoxic compounds. As said, the assay consists of 6 different biomarker genes. There's 2 different biomarkers for the detection of DNA damage. A gene called BSCL2 that is activated upon the formation of bulky DNA adducts and inhibition of DNA replication. Typically correlates well with the induction of mesogenic DNA lesions. And the second marker is triggered by the induction of DNA double-strand break, so a chromosomal damage. There's a marker for the activation of the p53 response that is obviously related to the induction of DNA damage, but can also be triggered by other stressors, including oxidative stress. ToxTracker has 2 markers for the formation of reactive oxygen species and oxidative stress. There's a bit of crosstalk between all of these pathways, so that's why there's different markers for oxidative stress. And finally, there's a reporter gene for the unfolded protein response. To give you a quick understanding of how the assay actually runs, it's a 2-staged assay where in the first stage, for every compound that is tested, a broad dose range finding is performed to find the concentrations that are suited to test in ToxTracker. Typically 20 concentrations are tested. And after 24 hours exposure, the cytotoxicity is estimated by doing a cell count. And the advantage of this dose finding is that it also provides information about autofluorescence of the compound, metabolic activation, since the assay includes also S9, compound solubility. And in the second stage, then the 6 different reporter cell lines are seeded in 96 well plates, or nowadays we can also do it in 384 well plates for increased throughput. Typically 5 concentrations for each compound are tested in the absence and presence of S9, and after 24-hour exposure, fluorescence of the different reporters is measured using flow cytometry. And again, the cytotoxicity is estimated by doing a relative cell count. Just to give you a very quick example of what that looks like. So here you see 4 toxic compounds on the left. So cisplatin and etoposide are 2 genotoxic agents. And as you can appreciate, with an increasing dose, there's an activation in red of the 2 DNA damage reporters, classifying the compound as genotoxic. We would interpret this as that there's formation of bulky DNA adducts. Compounds are likely mutagenic. There's the induction of DNA double-strand breaks. So chromosomal damage, there's p53 activation, but very little oxidative stress. Compound number 3, diethylmaleate, selectively induces oxidative stress. As you can see in blue, the two oxidative stress reporters are activated. And also p53 is activated, but no DNA damage. And finally, tunicamycin interferes with proper folding of newly synthesized proteins, selectively induces the protein damage reporter. So this indicates the specificity of the system. System works with S9, liver extracts from rats, typically arochlor or phenobarbital induced rats, to include metabolism in the system. There's no time to go into details, but just as an example, aflatoxin B1 is not reactive in the absence of S9, but after the addition of S9, compound becomes genotoxic and also clearly cytotoxic. So I said the advantage of the ToxTracker system is that it can provide information into the or insight into the mode of action of genotoxic compounds, and I would like to show you 2 examples. Example 1 is an extension of the ToxTracker system where we, in addition to the 6 cell lines, also use information on cell cycle progression as well as polyploidy or aneuploidy of the cells. This extension we call ToxTracker ACE for antigen classigen evaluation. And just to give you one example, on the top here you see the classigen cisplatin. On the left in ToxTracker you can see that cisplatin activates both of the DNA damage reporters here in red. And in ToxTracker ACE this is combined with measuring cell cycle distribution of the cells after 4 hours as well as after 24 hours. After 4 hours, you're going to see that the cell cycle profile is not really affected by cisplatin. After 24 hours, it clearly is. But after 4 hours, there's no effect. After 24 hours, there is no induction of aneuploidy in the cells. But in contrast, if we look at, in this example, at tubulin poison Taxol, we typically see selective activation of only one of the 2 DNA damage reporters in ToxTracker, typically only the activation of the DNA strand break marker, although we do see that if you push the system far enough under high cytotoxic conditions, you basically see activation of many different reporters. But so to confirm that this is a non-DNA reactive substance, we include information on cell cycle progression, and now clearly after 4 hours, you see that cells start to accumulate in mitosis, observed in the cell cycle plot in the center, where the peak on the right highlights the cells in the G2 or M phase. And in addition, after 24 hours, there's a clear increase in cells with abnormal chromosome numbers, so aneuploid or polyploid cells. So in this way, the assay, the ToxTracker assay can be extended to directly differentiate between classogenic or antigenic agents. The second mode of action that I would like to highlight is the induction of oxidative stress and indirect genotoxicity. 2 examples. First example is potassium bromate. This is a compound that induces the oxidative stress response, clearly on the left activation of the blue reporters. There's also again p53 activation, but for potassium bromate we also see that the marker RTKN for DNA double-strand breaks is activated, suggesting that there's DNA damage being induced. Now, to answer the question whether the DNA damage is secondary to the oxidative stress, we can run ToxTracker in the presence of a Ross scavenger, typically N-acetylcysteine or glutathione. And in this example, you can see in the middle part that we have induction of the oxidative stress response as well as the DNA damage response marker. On the right of the graph, you see that when we add a Ross scavenger, in this case N-acetylcysteine, we strongly reduce the oxidative stress induction in these cells, and by doing so, we also lose the induction of the DNA damage reporter, strongly suggesting that the strand breaks are secondarily caused by the formation of reactive oxygen species. But as a second example, MMS or a methylating agent methylates DNA as well as proteins. Also here we see a lot of oxidative stress being induced, but also activation in this case, both of the DNA damage reporters. Again, when we do the same trick with N-acetylcysteine, again on the right you can see that that N-acetylcysteine almost completely abolishes the oxidative stress, but in this case the DNA damage response is not affected at all, suggesting that the DNA damage response and genotoxicity is rather related directly to the interaction of MMS to the DNA. So as many of you might have heard, ToxTracker is currently under evaluation by the OECD to see if ToxTracker can be used in regulatory applications as well. So this study is ongoing for more than 2 years now, meaning that there's 8 laboratories involved in testing ToxTracker internally and looking for its reproducibility and transferability. These are the labs that are involved. On the left, you can see the labs that are actually doing the work. The only note is that Kao Cosmetics from Japan during the project had to step down due to internal reorganizations. But overall, I think we have a good mix of companies from pharma, chemical, and cosmetics industries, and 2 large CROs. And in addition, we have an independent validation management team that consists of I think well-known experts in the fields. And so this project is ongoing at the moment. This is the status. So in total, 64 compounds will be tested in the study. Phase 1 has been completed, which was the technical training of the labs. Phase 2 has been completed, which was proficiency testing of the labs. And we are now almost getting to the end of the final phase, where each lab is asked to test 24 compounds. Most of the data is currently being analyzed, and there's a few loose ends, some retests due to technical inconsistencies. But overall, the project is at least the experimental part is getting to the end. So we are hoping that we can finish at least the first part somewhere in spring 2020. Although, of course, we have to see how things are progressing. So last 5 to 10 minutes before I'm going to transfer the presentation to the guest speakers, is to highlight a few things that are currently going on at Toxis. And there are actually 4 new things that I would like to share with you. And I will go through them one by one. And the first is a platform that we call ToxTracker Academy. We are receiving a lot of questions from people that are interested in the ToxTracker assay, and they want to learn how they can use it, and background information, or maybe look for events. And so that's why we started the ToxTracker Academy. And this is really a landing page and a continuing education platform where you can find all relevant information about the assay. There's information on the scientific background, on events and activities that we organize, as well as possibilities for collaboration. So the website is live now. So after this webinar, you can go and check it out. You will find our information on practical courses and events that we host, as well as information on the background of the assay. And just to give you a heads up, so we've scheduled 3 events that are related to ToxTracker. Next month, there will be a technical webinar together with Miltenyi Biotech. We are actively using their max quant flow cytometers, and in this good collaboration, we will have a joint webinar to show how the ToxTracker system works well with their flow cytometers. And there's 2 practical courses being planned now. The first will be happening in July. Obviously, if the coronavirus outbreak doesn't continue into the summer, we'll have a practical course organized here in Leiden in the beginning of July, and we'll also host an hands-on training and practical workshop alongside the Eurotox meeting in September, and that will be together with Lundbeck, So the labs of Lüneburg will be the site, and also Miltenyi will provide the materials. And on this website for the ToxTracker Academy, you'll also find some suggestions, ideas, and examples of ongoing collaborations. So the second, and this is a real new product I would like to introduce, is an extension of ToxTracker, where we have created a new reporter cell line. So in addition to the 6 reporters that we have right now, we have created a GFP tubulin reporter cell line, following the same technology as used in the ToxTracker system, so having physiological expression of an endogenously expressed GFP tag tubulin. We can use that to further investigate the mode of action of endogens, and obviously particularly focused at tubulin poisons. The principle of the assay is rather simple. So the cells express GFP tubulin, and after exposure to a compound, we can do a permeabilization of the cells and wash out the free GFP tubulin, then fix the cells and look for a decrease or an increase in tubulin. And on the lower right, you can just see an example where indeed, if you add a tubulin stabilizer to the cells, then you see an increase in your polymerized GFP tubulin signal. If it's a destabilizer, it's a decrease. And this is just an example of of 2 compounds, tubulin stabilizer Taxol. In the middle part, you will see that it affects the cell cycle progression. This is after 4 hours. You see that cells start to accumulate in mitosis. And then if we measure GFP tubulin signals, there's a nice increase in the amount of polymerized tubulin in the cells. And in contrast, if we treat the cells with nocodazole, tubulin destabilizer, then you see also here that that impacts the cell cycle progression, and cells still get arrested in mitosis. But now you see a steady decrease in the amount of polymerized tubulin, as you would expect. And we've been validating the system so far, and as you can see here, so what you see in the plot is the area under the curve in a dose response in tubulin signals. And you can see that for the tubulin stabilizer, there's a clear increase in GFP signals, where the tubulin destabilizers all result in a decrease in the GFP signal. And the advantage is that we can do this in living cells. So at any time, we can, during exposure, we can basically follow the dynamics of tubulin polymerization, depolymerization. So I think this makes it a very flexible system to dive into the mode of action. And obviously, it fits very well in an AOP approach, where you want to dive into the mechanism of an endogenic compound. Very quick slide. Something that we have been talking about for some time is our ToxPlot software. People that have been working with us probably know the heat maps that you get in the reports or have seen in publications. So we've been using ToxPlot to generate these heat maps. This ToxPlot always was an internal data analysis tool to take the cells from the flow cytometer and then analyze the GFP signals and plot them against cytotoxicity. But we have now expanded this ToxPlot software to a software platform that is usable by anyone. So future clients will have the opportunity to not only get a report, but also get access to their data in this ToxPlot software. So they have their own private access to their own data, can play around and have a look at the raw data themselves, can generate reports in the format that they would like. You can show it to your peers, you can show it to your management. You just have the flexibility to look and work with the data that we have generated. And this will be in addition to the reports and with the expert interpretation that we will be providing. And we'll be implementing this software soon. And the last thing that I would like to show you is a test that has been announced for quite some time. It's not really related to genetic toxicology, but this is a test for developmental tox. With our expertise in stem cell biology and reporter systems, we've been working for quite some time on a system that we can use to look for early stem cell differentiation problems and early embryonic development toxicity. And today we are now officially launching the assay that we call ReproTracker. This, without going into too much detail because there's no time. So this system is based on human induced pluripotent stem cells that we can differentiate into different tissues. Initially in early differentiation processes into ectoderm, mesoderm, and endoderm, and then we continue differentiation into mature tissues as a indicator for, let's say, maturation and early embryonic development. For now, we have the differentiation into functional cardiomyocytes as well as hepatocytes finished. We're almost there with also a neural tissue, so that's coming soon, hopefully, let's say, in a few weeks or months. Just to give you an idea about the protocol, again, there's a strong similarity to the ToxTracker system, of course. Also here for ReproTracker, we start with a dose finding, testing 20 concentrations, but in this case, look for the concentration where we start to see cytotoxicity. Obviously, if you go to high cytotoxic conditions, you will kill all differentiating cells. But that doesn't really indicate developmental toxicity. So in this case, we aim to find a concentration that at the top concentration should induce 10 to maybe 15 or 20% cytotoxicity, but try to stay in the range of non-cytotoxic conditions. Then we expose the undifferentiated pluripotent stem cells to 5 concentrations of a compound, and then in time differentiate these cells into functional hepatocytes or cardiomyocytes in protocols that run between 14 and 21 days. And during that process, we continuously monitor the differentiation of the cells by high content imaging and morphological profiling. And at distinct times, we isolate RNA from these cultures to look for the expression of different biomarkers. And then the system looks for disruption of, let's say, normal embryonic development or stem cell differentiation. And we can do that by applying different biomarkers. So on top you see, let's say, the undifferentiated cells, and on the right, the differentiated cells, where in green there's a staining for albumin, showing that most of the cells are indeed producing albumin, which is common for liver cells. And on the bottom, you see some of the biomarkers that we use to monitor the differentiation process running from undifferentiated cells through the early endoderm into mature hepatocytes. And we do the same for cardiomyocytes. They, after 14 days, they start beating. So we have a very almost confluent beating cardiomyocyte population that stains also for cardiomyocyte proteins. And also here, using different biomarkers, we can in time follow the differentiation of these cells. I don't have time to go into all the details. We have a poster that we prepared for the SOT, so we can share that with you. Just to give you 2 examples, a teratogenic agent, hydroxyurea. We identified a concentration we wanted to test in ReproTracker. Difficult to see here on the graph, but we look for morphological changes in these cultures, as well as in this case, I included only the 2 biomarkers that are expressed in the differentiated tissue. And as you can appreciate on the right, there's a clear decrease in expression of the markers for both hepatocytes as well as cardiomyocytes. And in all cases, the signals go below a threshold. Thresholds are set based on the standard deviations of the untreated controls. And so for this reason, we classify this compound as teratogenic. And the bottom example, amoxicillin, antibiotic, not cytotoxic, tested up to a rather high concentration. We, for this compound, did not see any morphological changes, but also in this case, we did not see a significant decrease of reporter activation after exposure. So therefore, this compound was classified as non-teratogenic. And so this is just an overview of some of the validation work that we've done. On the left, you see the table with in vivo teratogens. mostly, of course, based on the DART studies in rats. And you also see the results in ReproTracker. And for most of the compounds, or nearly all of the compounds, we also classified these compounds as teratogenic. So, and I have to say that this study was all done blinded, so the scientists did not know what they were testing. There are a few green boxes indicating that they were missed. For one or two, you could argue that we should have tested at a higher concentration, so there will be some work ongoing. We did notice that hormones are typically not so easy to identify in the system, possibly related to the lack of expression of certain receptors. And also DNA damaging agents were difficult to classify as teratogenic, mostly because these compounds are highly cytotoxic, and on non-cytotoxic conditions, there was no effect on the cells. So there are some limitations. We've also tested a number of non-teratogens. There we see that many compounds indeed were tested negative in ReproTracker, but we did see some of the compounds reported positive, so classified as teratogenic in ReproTracker. So we're still exploring that. For some of the compounds, we have indications that there's indeed differences in species. In vivo studies have been done in rat, and ReproTracker is based on human cells, so it might be species specificity. It might also have to do with the cutoff for cytotoxicity. So that's something that we are currently still exploring. But with the validation work that we've done so far, we are fully confident that in many of the cases, or nearly all of the cases, we are able to identify teratogenic agents. And if you want to learn more about this assay, feel free to contact me or Paula after this webinar, and we are definitely able to share the poster that we prepared for the SOT meeting. Just to close up, just a few practical facts about ReproTracker. So we typically test to a maximum concentration of 1 millimolar or 1 milligram per ml, which means that we require about 50 milligrams of a compound for a full test. Turnaround time of the assay is 9 to 12 weeks. It just takes 3 to 4 weeks already to test, to run the differentiation, and we run 3 independent repeats. So we require a bit of time. We can test up to, let's say, 4 to 8 compounds per week. You work with different solvents in the system, and the protocol is based on, let's say, a single biological exposure, but we start with a dose range finding where 5 concentrations of compounds are tested. We have positive and negative controls in the system, and for each compound we run 3 technical repeats. So with that, I would like to close this part. I suggest that we can take a few questions before we go to the next speaker.
Yes, so thank you very much, Jill. So for everybody, you can now use the chat function to ask questions if you like. We already had a few questions coming in. So Jill, can you say something about the typical positioning in a testing strategy for ToxTracker?
So that is a difficult question to answer, because one thing that I learned over the last years is that every user, both from industry or research institutes, see different applications of the test. The obvious, of course, positioning is as an early screen. I would say ahead of the let's say full GLP regulatory tests. Uses a very early ranking tool, or as an alternative maybe to the let's say flow micronucleus test, so really early screening. But we regularly get questions following, let's say, the in vitro GLP studies, Ames test, micronucleus test, where positive results are generated. And then the question comes, can we understand the mechanism of action, and can ToxTracker there help to figure out what's going on, and maybe to de-risk some of the compounds. But we've even done some tests following in vivo studies. So I think it's a wide range. It depends on the application and the questions you want to ask.
Okay. And then one of the questions I think many people have are regarding the timelines for regulatory acceptance.
That's another nice question. So although I would not say I'm an expert, I do see that typically the timelines for getting a new test into the regulatory space takes a lot of time. The OECD validation for ToxTracker is currently running. I'm very positive that we will be able to complete that study this year, and then we'll have to go back to the OECD and take the next steps. I think obvious steps would be to work on a DRP and investigate the positioning of the system, and at some point work on a draft test guideline. But I would like to emphasize that we already now see that many people use information from the system in their regulatory dossiers. Obviously not to replace any of the standard assays, but as a weight of evidence, additional information, explanation for, let's say, either positive or equivocal results. So there is a number of examples, and some we can actually share from applications where people use ToxTracker information in their dossiers. And of course, this is in line with the ICH guidelines, where that you can basically all scientific information that is used to build your case is acceptable. It's just at this stage not able to replace any of the standard assays.
Okay, well, thank you so much, Jill. I think we have to leave it at this for now, but please do send in your questions via chat and we will answer them later. I think by now we will go to the presentation from Jax from Rhythm.
Thank you, Paula. Can everyone hear me okay?
Yes, very well.
Okay. Thank you. Thank you, everyone, for joining today's call, and thank you for preparing the ground for me, at least, to go over the talk. So for today's talk, I'll be touching upon a case study of how we could leverage the utility of ToxTracker and redefining the GTOX animal alternative testing strategy. So as a traditional genetic toxicology paradigm, we try to address two different endpoints for cell density and class density. There are regulatory approved assays which are in vitro and in vivo. The upper part in the green box is in vitro studies and the latter part is in vivo studies. Depending on the outcome and the regulatory restrictions and or requirements, certain times in vitro studies have to be followed up by in vivo studies, and due to some restriction, there does not have to be followed up into in vivo studies and has to be stated as animal alternative space. For today's presentation, I'm going to stick to the right part of the screen right now, which is gastrogenicity assay. The case study that I'm going to present is on one of the fragrance material, which is 2-octene-4-one. This material, as you can see from the structure, is an alpha-beta unsaturated ketone. For this particular chemical, we did the traditional in vitro battery, such as Ames and in vitro macronucleus study, and the results from the Ames study was negative. However, there were positive responses observed in the 3R arm with and without S9 condition when we conducted a traditional in vitro using human lymphocytes. Considering this material was both flavor and fragrance, due to flavor requirement, there was a follow-up in vivo study conducted, in vivo micronucleus study, which was negative. So with this data set in hand, we were wondering why there was an in vitro micronucleus positive response and in vivo was clean negative. What could be the potential mechanism that could lead to this in vitro positive outcome? And it's clear in the in vivo study, which is considered as more biologically relevant system at this point. So we decided to leverage the use of ToxTracker assay to answer this question. And from the results of the ToxTracker assay, as you can see on the screen right now, there are more reporting strategies such as heat map and everything, but for today's presentation, I have kept it simple. If you look at the data table, the DNA damage marker, there was no DNA reactivity identified by ToxTracker at all. There was no protein damage and no reactivity as well. The main mechanism identified was oxidative stress and or cellular stress. For fragrance domain, unfortunately, after the SCC's 7th Amendment, which got into effect in 2013, the animal testing is banned. So to address the classificity endpoint, after we do the in vitro micronucleus study, and if it comes out positive, we have very limited options. There has been a few other models that are still under validation phase, such as Henneck micronucleus and 3D skin micronucleus, which could be utilized as a follow-up animal alternative testing strategy if we come up with the traditional in vitro positive outcome. But there is still a dilemma, which is the ideal system. How do we pick the system? Because we have two options here. So ToxTracker also will help us in identifying and deciding which would be the appropriate system. So right now we have mechanistic information from ToxTracker assay, the traditional in vitro micronucleus, which is oxidative and cellular stress. What if we don't have luxury to conduct an in vivo study? And what if the in vivo data didn't exist in this particular case? I decided to run an in silico prediction tool to identify which could be the potential metabolite and target, and what could be the mechanism of action. By the prediction tool, it will identify the oxidative and cellular stress could be created due to Michael addition due to activated double bonds. And both these chemical structures you can see on your screen are eliminated by glucuronidation pathway. So based on this information, considering 3D skin model has sufficient glucuronidase activity, it could be considered as an appropriate follow-up. And also, it is a primary route of exposure for fragrance materials, so it is more ideal testing system. Now, if the target and or metabolite identified by in silico with the mechanistic information from ToxTracker predicted it to be a sulfotransferases, in that case, headache micronucleus could be considered as an appropriate animal alternative testing methodology. So we decided to run a 3D skin micronucleus study, which was negative. So if you see, it's consistent with the results observed in the traditional in vivo micronucleus study as well. This could potentially help us eliminate animal testing and give us what could be the appropriate follow-up study and lead to less false positive. So just to brief it up, in the classicity data set, if we decide to run in vitro micronucleus study using lymphocytes, if it's negative, there isn't any issue. But if we end up with a positive response, then we decided to run a ToxTracker assay, which will help us identify what is the potential mechanism of action leading to this positive. And with the help of ToxTracker in conjunction with it will also help us determine which will be an appropriate animal alternative follow-up testing strategy. With that, I would like to conclude my presentation, and I would like to acknowledge the Vice President of RIFM Research Program, and Marie Happy, as well as RIFM core team, which comprises of experts from the industry, and the adjunct team, which comprises of the experts from the academia setting and our research organization. Just a food for the thought. If you see the soap, please use it. At least 20 seconds is recommended. Social distancing is a global champion. Stay safe, everyone. Stay safe, my friends. At this point, I can answer any questions, provided it's not related to when is the market going to go up, because I don't have answer for that. Thank you.
Great. Well, thank you so much for this presentation, Jax. There are a few questions. One of them is that oxidative stress plays a role in for some of the genotoxic effects that you see. Do you think that a Ross scavenger assay can be of added value there?
So that's a good question. I was thinking about that when I was listening to his presentation as well, that we should follow this particular chemical with the Ross scavenger as well. As of now, we haven't tried that approach, but that's a good point. We might consider that in future if the identified mechanism is oxidative and or cellular stress.
Okay, thank you so much. Let me see for the questions. Yes, we have a question. If you can tell something about the correlation between ToxTracker and the 3D skin micronucleus.
So correlation as in, if you are asking about the positive and negative rate, it's still to be answered, but ToxTracker definitely helps us pick the right testing system once we have a traditional in vitro battery positive. As of now, we have run more than 15 chemicals in ToxTracker as well as 3D skin model. We have the data back as of last week, but we are still evaluating it. Once we have that data and information available, we'll share it with the audience via either poster presentation or some other media or any talks.
Okay, Perfect. Well, thank you so much for your presentation, Jax. As said, the rest of the questions we will leave to after the presentations in the end. So we're going to the final presentation by Raja from Corteva Agrisciences.
Thank you, Paula. Are you able to hear me well?
Yes, I am. Thank you so much.
And are you able to see my screen?
Yes, you can.
Okay.
You can go ahead.
Okay, perfect. Thank you. Well, good afternoon, everyone. Thank you for everyone for joining this webinar. So in this presentation, Shreel asked me to give a brief presentation on how we are applying ToxTracker assay for assessing genotoxicity potential of agrochemicals. In this presentation, I will provide a brief overview on why we were interested in the ToxTracker assay, how we are applying this assay, and some of our key learnings. Considering the audience, I wanted to give like one slide, very brief overview on the testing requirements for agrochemicals. So in general, in the ag sector, assessment of mammalian toxicity profile is a very important component of standard safety evaluation process. So our products need to be safe, not just for humans, but also to the environment. And this extends to, for example, consumers, farmers who will be using these products on a daily basis, employees in the manufacturing, transport, pets, wild animals, birds. From a mammalian tox perspective, pesticides have most comprehensive data requirements. A product in general would undergo greater than 100 rigorous studies to support the health, safety, and environment that is required for registration of our products globally. Genotoxicity is one of the most important endpoints that we assess as part of the mammalian toxicity assessments. So as part of this, each active ingredient, formulation, plant or soil metabolites are evaluated for mutagenicity, clastogenicity, and anigenicity using a battery of in vitro and in vivo studies. The boxes I've included here, for example, for each active ingredient, the initial basic battery of studies would include AIMS, in vitro mammalian mutation test, in vitro micronucleus test, and in vivo micronucleus test. Likewise, I listed the studies that are required for plant or soil metabolites and formulations. So the main purpose of this slide was to highlight the point that even from GeneTox standpoint, we do conduct a number of studies to assess safety. When we look at the development of a new active, so in general, it goes through a stage gate process, and each active may take a little over 11 years to complete this process. The stage gate process would include, at a very higher level, discovery phase, pre-development, and development phase, where decisions are taken at various stages to determine if that molecule should be moving forward or not. Most of the standard GLP studies, here I highlighted only the GeneTox studies considering the purpose of this presentation, but most of the GLP studies are conducted during the pre-development phase. And majority of the studies that are conducted during the discovery phase are screening type in nature, where the need is to identify major red flags at the earlier stages of a molecule development. One of the major challenges that one would have at this discovery phase is that the amount of test material that is available would be substantially small. So as a result, we would need an assay that requires smaller amount of test materials. Speed is obviously a very important criteria during the discovery phase, and another more important criteria is accuracy, sensitivity, and specificity. So this is where ToxTracker fits the bill for us. The major advantage we saw with the ToxTracker was the mechanistic information that it provides, both from genotoxic and non-genotoxic endpoints, as is shown by the cartoon here on the right side, and Phil described it very well in his presentation today, and also a number of publications. And Maria Donner in our company was played a huge role in having this assay in our company. Other major advantages that we saw with the ToxTracker assay was like very high sensitivity and specificity, as highlighted by the numbers here. Smaller test material requirements that is needed for this assay, and much shorter timeline compared to the standard GeneTox assays. So as Sril mentioned, we were part of this international OECD inter-laboratory validation. We were one of the 8 or 7 laboratories who evaluated or tested a number of compounds in the ToxTracker assay. So the process included phase 1, which included technical training by Toxis group. I would really like to appreciate and thank Shreel and also Remco Der, who visited our site at Newark, Delaware, and helped our folks get trained on this assay. So as part of this testing, we tested 8 reference compounds, both in absence and presence of S9, 3 independent replicates. Phase 2 included blinded study testing 6 compounds, both in absence and presence of S9. Both these phases were complete last year. And the third phase included testing 24 blinded compounds, both in absence and presence of S9. So as part of the study, we conducted or tested 8 compounds per each run. All testing was done in triplicates on independent days within a week. And we haven't seen the results yet, but based on the recent discussion that we had as part of this validation study, we were notified that 21 out of these 24 compounds met the criteria. And for the remaining 3 compounds that did not meet the criteria, based on my understanding, the main reason for this is not much to do with the assay or the technical aspects. It is primarily because of discrepancy in the cytotoxicity assessment between the pre-tox and main run. So in this study, just like other GeneTox studies, there is a pre-tox study where a compound is tested at 11 different concentrations, and the highest concentration is selected based on cytotoxicity and precipitation. So in these 3 compounds, what happened was so in the pre-tox run we saw higher cytotoxicity, but in the main run we did not see that cytotoxicity. As a result, we may have to repeat the remaining 3 compounds. And the cartoon on the table that I'm showing on the right side shows our positive control data, where this column shows the cutoffs for calling a positive as positive, and the last column shows our responses, indicating that in general the positive control data passed very well, with the exception of aflatoxin B1, where the value is 1.8 versus the cutoff is 2, and I have a slide later to talk more about it. So how are we applying the ToxTracker assay? So early stage screening is like based on the introduction that I provided. This is obviously the place where we decided to use, especially considering the amount of test materials that are required for the GLP studies. ToxTracker would definitely fit the bill. And the way we are interpreting the data, so we just started using ToxTracker assay for our early stage screening for about like 3 to 4 months back. So I don't have a lot of data to share, but I do have some data that I would be presenting. But the interpretation criteria that we have or that we are using now is that, like, for example, in the ToxTracker assay, if all our biomarkers come out negative, or the biomarkers for mutagenicity and DNA strand breaks come out as negative, then we would ensure that the molecule proceeds further in the pipeline. If the mutagenic or DNA strand break results are equivocal, then depending on the results that we get here, we may consider conducting screening aims and or screening in vitro micronuclear studies. And if the results are clear negative, then the molecule would proceed further in the pipeline. Otherwise, the molecule would be stopped. Similarly, if a molecule is positive, both for mutagenicity and DNA strand breaks, then that molecule will not proceed further. Other application, how we could use ToxTracker is for mechanistic information. Sheel mentioned it in his previous presentations. For example, if we have a equivocal or positive result in the micronuclear studies, then one could conduct the ToxTracker study to determine if the positive response is because of interaction with the DNA, or in other words, is it because of DNA damage, or is it secondary to some form of stress? I think the ToxTracker would play a huge role in understanding mechanisms or reasons for a positive response in the micronuclear studies. Recently, in some of our routine screens, we came across a set of compounds that were found to be positive for aurora kinases. And based on the existing literature, we know that aurora kinases are critical for proper passage of cells through several stages of cell cycle for cell division, especially in mitosis. And inhibition of aurora kinases could lead to aneuploidy. And the table that I'm showing here, I got it from Tang et al. 2017 paper. It shows aurora kinase A, B, and C, where it is localized, and the main functions, which are mostly around cell division in the mitosis phase. And there are a number of publications which shows that perturbation of or like aurora kinase binders or binders would have a significant impact in a number of tumors. And the cartoon on the right-hand side shows the cellular localization shift of aurora kinases in mitosis. Here, I apologize, it is a little difficult to see the color contrast, but basically the cartoon indicates how essential the aurora kinases are in the standard cell cycle. Coming to our data, the table on the left-hand side shows results from kinase assay screens, where I'm showing here data for 3 compounds. KD values, which would stand for equilibrium dissociation constant, which basically shows about the affinity of the compounds have towards the aurora kinases. The smaller the value here, the stronger the affinity is. And the figures that I'm showing on the right-hand side are the results from the ToxTracker A's. The y-axis here is percentage of aneuploid cells. X-axis is concentration in micromolar for all 3 compounds. And as we can see, so in ToxTracker, all these 3 compounds were identified to be aneuploid. And there is a decent dose response in the aneuploidy, although the dose response is not very strong, but there was a decent response and it was positive for the aneuploidy. So in general, there was a good correlation between the selected aurora kinase binders and anagenicity. In the next 3 slides, I'll present our experience or key learnings working with ToxTracker thus far. First of all, the cells are very easy to maintain. Procedures are very clearly described in the SOP. The data that we generated in-house is consistent across independent replicates. Smaller test material requirements, that was a huge advantage for us to implement it during the earlier stage of molecule development. One of the great advantages that we saw here was the technical support that we received from Toxis. Like very quick and open communication. I would really like to thank Toxis team for this. It was really, really helpful. Other experiences. So in general, the people standpoint, we do need to have, it is not as resource intensive as, let's say, for example, conducting a HPRT study, but it does require multiple people to perform the assay. So here I'm referring to, let's say, like we typically test 8 compounds per week. So the numbers that I'm showing here is based on that criteria. And even from the flow part, we use Canto flow instrument, and it takes about 32 minutes. So in a typical day, we would spend about 8 hours for this experiment. So again, I think maybe 6 months down the road, when we have more and more experience with this assay, maybe I wouldn't see this as a huge challenge, but at least for beginners, this is something that they need to consider. And again, here the support that we received from Toxis has made the process really helpful, really smooth actually. And with regard to the ACE data generation, so the ACE part, it was not part of the interlaboratory study, so it required some additional training, and we needed some time to reduce well-to-well variability. There were so the time was spent to like ensure some of the technical procedures are being conducted to reduce the variability, creating the templates for gating for the flow cytometer, macros enabled data analysis sheets. Here again, I think I would really like to thank Inger from Toxis, who was helpful to put together all this information. And I hope that the Tox plot that Shreel mentioned earlier would include some of these macros enabled sheets, which would make the process much smoother for new users. And another thing that we worked recently as part of the ACE data generation is to develop our own historical negative control data, as this information is required for calling a chemical positive for antigen-ST based on the foreign population. We also had some challenges with aflatoxin B1, which is a positive control. We had challenges to have a positive result on a consistent basis. So the table here shows aflatoxin B1, and the red boxes indicate where we had challenges meeting the twofold criteria. So after discussions with Toxis, we chose cyclophosphamide as an alternative for our internal testing, and we also decided that we would apply the same criteria for cyclophosphamide as we would for aflatoxin B1. And currently we are testing both in conjunction, and in future we plan to move to cyclophosphamide. And here again in the table, the cyclophosphamide retamine, we had no problems meeting the criteria. So in conclusion, the assay has higher sensitivity, specificity, and accuracy to identify genotoxic compounds. Very good performance in interlaboratory studies. From our application standpoint, as I mentioned, we have been applying this assay for early stage screening. And this is one assay that we are looking forward for mechanistic assessment for as a follow-up for our positive micronucleus studies. I'd like to acknowledge Stephanie, Abby, and Freedom. They generated all the data. I'm just speaking on their behalf. And I'd like to thank once again Toxis team for all their help and support in this journey. With that, thank you. And if there are any questions, I'd be happy to address.
Yeah, so thank you very much, Raja. There are a couple of questions. One of them is, what are your considerations for running the assay in-house instead of using it as a service?
Yeah, that's a good question. So we do have a lab in-house, and we do conduct majority of our gene tox studies in-house. So the main advantage we see with it is, in general, I'm not talking only about ToxTracker, but also in general having a lab, right? I mean, we would have better control over the study initiation and also communication during the studies. And as we were part of the interlaboratory studies, specifically on the ToxTracker, we had the training and experience to conduct the assay in-house, and we had all the required instruments and people resources. And also another main reason being, in general, it becomes a little challenging for companies to send their very early stage molecules outside the company. So if we do have a lab, then in general it becomes a preferable approach to do the testing in-house. So basically, it's a combination of all these things.
Yeah, sure. I can understand that. So thank you. One of the questions that came in is how critical will the historical control value play in the interpretation, and what is being done to generate this data for both GLP and non-GLP assays?
Yeah, that's a good question. Now, like historical control data is something that I mean, in the again, I think going beyond ToxTracker, in general, gene tox, endocrine studies. This is one area where a lot of emphasis has been put recently, coming from the regulatory agencies and also even internally. I think it becomes important to understand what the level of variability and where our values, negative control values stand. I think it's really important to understand that. So ToxTracker, we are primarily conducting non-GLP at this stage. And what are we doing to generate the data? So basically, we are ensuring that we would have extra negative control plates every time we do conduct the ToxTracker assay. So we are putting considered effort to generate this historical control data in-house, because as you would expect, right? I mean, like toxes have historical control data, but again, um, the values are bound to change because of various technical reasons. So we are making sure that we develop our own historical control data, especially for the anigenicity part.
Okay. Thank you so much, Raja. One of the notes I want to say to all of the attendees, if you want to ask questions, could you please use the chat function or the questions function? I already see some questions coming in, but I also see some raised hands, which do not allow me to bring up your questions. I think one of the general questions that we received, let me look up the chat, is a question on if the compound is positive for oxidative stress and positive in the in vivo micronucleus assay, what follow-up assay could you run? And maybe this is also something that Jill has some input in.
Of course, it's a good question. A part of the answer probably lies in the chemistry that you're looking at and the information that you already have on the compound. Typically, we see that compounds induce oxidative stress but are not DNA reactive, that they are typically negative in vivo because normally oxidative stress typically occurs at rather high concentrations because you first need to saturate the antioxidant systems in the cell, and those concentrations you typically do not reach in vivo. So we very often see examples where in vitro micronucleus is positive, but then if we identify oxidative stress as the primary mode of action and we don't see any indication for, let's say, direct DNA reactivity, these compounds are then negative in vivo. If you do get an in vivo positive, It's a good question. Maybe the other speakers can also suggest it. I mean, I would definitely not suggest to run the COMET assay, because they will also be positive by my prediction. Obviously, I assume that an in vivo mutation assay has already been performed, but otherwise that would be my first next step. And then it's a matter of, can you build a case where you would argue that there's a threshold. So it also depends on the concentration where you start seeing your micronucleus positives. But in vivo positives, you probably need a good dossier to explain the mode of action if you want to get around that. And for sure, your mutation assay should be negative. But I don't know how either Raja or Yax feel about that. Maybe you can, Raja, can you comment on that as well?
Sure, yeah. So I think if oxidative stress is the primary reason, if it is negative for mutagenicity, then I think this is where we would typically also need to take into consideration the exposure scenarios, right? I mean, like in our gene tox studies, we do test at very high concentrations. One question that we need to ask ourselves is, like how relevant also are those results in a real life situation? In general, the antioxidant system, as Phil, you mentioned, right? I mean, like it needs to be really overwhelmed for one to see purely oxidative stress related carcinogenicity, for example. And such instances are very rare in the literature. They do exist, but very rare. So in my opinion, I think we should look into the, like the exposure, real life exposure scenarios as well, bring in the biological context when interpreting such data.
Yes. Raja, do you have anything to add to this discussion? Oh, sorry, Yax, do you have anything to add to this discussion?
So, yeah, I would agree with Raja in this case that I haven't seen many chemicals practically which has been shown to be oxidative stress and has resulted in a positive outcome in vivo studies. But there has been few cases in flavoring industry, specifically EFSA has discounted some of the studies based on glutathione depletion issue or glucuronidation depletion issue as well. One of the other route is to try and do N-acetylcysteine, which can or NADPH, which will help regenerate glutathione. That's one of the options, or try ToxTracker along with N-acetylcysteine first, and then go to in vivo with the same approach.
Okay. Thank you so much, Yax. We had another question coming in. In case you can attribute positive findings for classigenicity to oxidative stress, can you claim a safe threshold?
Difficult question. I'm not sure that I'm the right person to answer that question. It is this is typically a question that you would ask in a, let's say, for a regulatory application. Of course, if you see a high level of oxidative stress and that results in DNA strand breaks, I think it is unfair to call the compound safe. Then if there is a threshold, that I think depends on case by case. I think it's too simple to answer that. But it also probably depends a bit on the regulator as well as the chemical space that you're in, whether you're talking about a pharmaceutical versus a fine chemical, because the regulations also differ a bit. Don't know if the other speakers want to comment on that.
Okay. Then I think we will go to a final question. The question was, the in vitro assays provide a caveat in terms of solubility. However, the in vivo assays, there is no such parameter. Is there anything being done for this aspect for the ToxTracker assay? So perhaps Jill, you're the best one to comment.
Yeah, solubility is always a challenge. For the ToxTracker system, there have been clear criteria set for acceptability of data, and that also relates back to solubility. Actually, this was one of the findings that came out of this inter-laboratory validation, where it turned out that the definition that people had around solubility differed a lot. And so we were forced to set very strict criteria. We always limit the maximum exposure to the maximum soluble concentration. Of course, some and that works, I think, reasonably well. For instance, for pharmaceuticals, some of the materials, especially chemicals, sometimes the material itself is not soluble, but is slowly dissolving or leaking, or even particulate matter requires testing. And for the different materials, we do have different approaches, but we typically, of course, are limited by solubility. And it's very difficult to translate that back to in vivo. We do occasionally, let's say, have modified protocols to work on, let's say, poorly soluble compounds, either by using different solvents or making sure that you look for, let's say, a migration of substances from either an inert material or into a solution, either into cell culture medium or in an organic solvent. We typically also, where possible, try to track the amount of soluble material in the medium, if that's relevant, and sometimes even try to track how much is getting into the cells. So we're trying to get our way around it, but solubility, of course, also in vivo is a question, but it's not an endpoint or a parameter that is typically addressed. But in that, I mean, in large sense, the honest answer is that we try to work and to get protocols into place to work with soluble as well as insoluble materials. But to some extent, the assay is not different than any other in vitro system. So defining what you're testing, I think, is the best approach here. And we try, I think, always to do our best to see if we can understand what we're actually measuring.
Okay, thank you so much for your answer, Gil. I think with this last question, we will conclude this webinar. One of the things we've noticed by email is that some of the people had problems, technical difficulties to get into the webinars, probably likely due to capacity. We would like to inform you that the webinar has been recorded, and we will see whether we are able to share the webinar afterwards. So we will get back to you on that. I will make sure that all the questions that have not been answered now will receive a private answer from us. And I think everybody has had the email by me. So if you have any questions after this, please feel free to contact me. And with that, I would just like to thank you for your attendance. And I hope this webinar was informative for all of you. Thank you so much.
Thank you.